All right, part 2 of your multiple choice for your final exam. Genetics. You need to know a couple of keywords. These are basically like vocabulary words. Can you pick them out? Homozygous means you have the same alleles. An individual that is heterozygous has different alleles, and your phenotype is the appearance of the organism. Now, I don't have any kind of crazy crosses on this one as I did on the other one, so it's basically just multiple choice picking out answers, but you have to use your knowledge to do so. So you need to know about monohybrid crosses and percentages of offspring. So if I would say you have an individual that's like heterozygous for a trait and an individual that's homozygous dominant for a trait, what percentage of offspring would be exhibit the dominant trait? You'd have to be able to figure that out. Or if I would give you 50% of the offspring are heterozygous and 50% of them are homozygous recessive, what are the parents? So you've got to be able to work forwards and backwards in that monohybrid crosses. You need to know that the P generation are the parent generations, and that's typically the pure breeding ones. So they will give you like all white or all purple flowers. And then pure breed plus pure breed gives you heterozygous in the F1 generation, and those will display the dominant phenotype even though they carry the recessive phenotype. You need to know that dihybrid crosses of heterozygous individuals, and I've got examples here of what those heterozygous individuals' genotypes would be. Anytime you have heterozygous plus heterozygous and a dihybrid cross, you will always end up with this 9:3:1 phenotypic ratio. And if you can remember that, you'll get that question right on the test without having to work out the full Punnett square. Otherwise, you're going to have to work out a 16-part Punnett square in your test, and that sucks. We don't have time for that because there's like 82 questions on this thing. Law of independent assortment states that genes on different chromosomes assort independently of each other in meiosis. That's just one of those fact questions. If I give you a parent's genotype, what possible alleles do they have? So if I give you a genotype that's this, what alleles could that parent make? Well, they could give this dominant gene or this recessive gene allele. And write a genotype given phenotypes and allele choices. Now, you had a really long one on your study guide last time, and you had a long one on your test. It's that same kind of question, just with a couple of different words inserted. So you should be fine for that. Other inheritance facts, you need to know that the sex-linked traits in humans is mostly on the X chromosome because the X chromosome has like twice as many genes on it as the Y. And you need to know that color blindness, of course, is on the X chromosome. If you are a carrier female and you mate with a colorblind male, which would be, I'm erasing, try again. If you mate with a colorblind male, you look like this, or if you mate with a normal male, you look like this. You need to be able to do those Punnett squares and figure out what the probabilities of your children, whether it's a female or a male being colorblind, would be. So make sure you can do all that. Meiosis, you need to know that it has 2 divisions. In prophase 1, it does this really special thing called crossing over. It's where the genes exchange their genetic information. You need to know that if a germ cell has 10 chromosomes, then there are 5 homologous pairs, right? Number 1 from mom and number 1 from dad. Then after the DNA replicates in the S phase, it will have 20 chromosomes total, but it will have 10 chromatids, because remember, the chromatid are these chromosomes attached at the centromere. So this is one sister chromatid, but it's full of 2 chromosomes. You guys did really well on that question on your regular test, by the way. After meiosis, there are 4 chromosomes in each sperm. How did I get 4 from 10? Goodness, I didn't proofread this very well. I was just trying to get it done. That should be 5 chromosomes because 5 is half of 10. So catching my mistakes here before I put this online for you. You also need to know that if a diploid cell has 10 total chromosomes, then it will have 5 types of chromosomes because it will have 2 of each type. Again, that's 1 from mom, number 1, 1 from dad, number 1, 1 from mom, number 2, 1 from dad, number 2, and so on. If it's normal. If it's not normal, then it could be anything, any combination. It might not actually be a functioning cell. DNA, the real simple base pairing rule, A pairs with T, C pairs with G. You're going to have to know the steps of replication. So the first thing that happens, of course, in the bad pickup line that you guys know, is helicase will come and unzip the DNA. After that, polymerase will bind to the DNA and start to add the bases, and then ligase will come through and join the fragments at the end of it. DNA will always begin at a replication fork. Nucleotides are always added at the 3' ends. Remember, the house always has to be pointed up. It uses RNA promoters to attach to that DNA to give the DNA bases something to pair to join to as it starts its chain. And then the sequence of the new strand is the complement of the parent. It's not the exact same as the parent. So make sure you know that that's a difference. And one strand is always lagging and is made in fragments. Those are those Okazaki fragments in the strand that's having to, it's the 5 to 3 strands, or sorry, it's a 3 to 5 strand, so you're having to lay down backwards as you go. Okay. Gene expression, we're almost done. So this is the new material. Actually, I guess some of this DNA from here on down is new material. But gene expression is what we covered mostly in this week and last week when you had Mr. Johnson. So chromosome has genes, and genes are made from DNA. Those are those big ideas. RNA polymerase is going to bind to a promoter on DNA to cause that transcription. Remember, transcription is making RNA from DNA to occur. There's a difference between RNA and DNA. There's actually a couple of differences. RNA has this uracil, and it does not have thymine. RNA is always single-stranded, so it will never have a complement. Well, in very rare cases, which you'll hear in the notes later, it has a complementary strand, but that's very, 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 very unusual. It's a type of, don't worry about it right now. Okay, so RNA is single-stranded, and it uses a 5-carbon sugar instead of the 6-carbon sugar. That's the ribose instead of deoxyribose. Translation is when you take that RNA that you just made and you make a polypeptide chain from it, which is your protein. If you have mutations, remember deletion is when you remove part of a chromosome, inversion is when you flip part of a chromosome, insertion is when you add a chromosome or add a base in place. Translocation is you take one chromosome and stick it on another one, and then substitution is when you can move one base pair for another, and A would replace a C, for example. So all of these can have issues or be silent depending on how it happens and how you inherit them. Like inversions and translocations are chromosomal. Insertions can also be chromosomal or DNA related, but deletions and substitutions are where base pairs are changed. There are main steps of gene expression and how they go from DNA to proteins. So the first step is DNA is used as a template to make the pre-RNA, pre-mRNA. Then remember it's processed, you get the poly A tail, and then the end cap, you get RNA to leave the nucleus next. Finally, that plus the ribosome is brought to the endoplasmic reticulum where this protein synthesis will take place. You add the polypeptides on, and the ribosome will then translate the mRNA and make the protein. So a lot of the newer stuff on how gene expression actually occurs is not on your final test. You don't have to stress about all the details from this week, so that's really good. Like I said, you can put this week's off until after you take your final if you want, because it won't be due until December the 14th if you're Monday, Tuesday, or December the 17th if you're Thursday or Friday. So happy studying. I'm going to give you your matching parts because the multiple choice is so long on the next part of this with the short answer videos. So I'll get those coming up soon.